Let us talk about how neurons communicate and send signals. All plasma membrane produce electrical signals by ion movements, and this is called the membrane potential. The difference in the electrical potential or the electrical charge between the inside and the outside of the cell is measured in volts. The membrane potential can be broken down into three main groups, and that is the resting membrane potential, a graded potential, and an action potential. The resting membrane potential is the membrane potential of a resting cell. The typical value for the resting membrane potential is negative 70 millivolts. Graded potential is the temporary localized change in resting potential which is caused by a stimulus. And an action potential is an electrical impulse that is produced by a graded potential, and it will propagate or travel along the surface of an axon, and it will cause the synaptic terminal to release a chemical called the neurotransmitter. Sodium and potassium are the primary determinants of membrane potential, and they can either be passive or active. When we say passive ion channels, they are also referred to as leak channels, and they are usually always open, and their permeability can change with conditions. Active ion channels, on the other hand, is also known as gated ion channels, and they open and close in response to stimuli. When the neurons is at resting membrane potential, most of these active ion channels are closed. This figure represents the four major types of ion channels in neuron: the leak channel, the chemical gated ion channel, mechanical gated ion channel, and voltage gated ion channel. I've mentioned that passive channels are also referred to as leak channels, and active channels are gated ion channels: chemical, mechanical, and voltage gated ion channel. Let us first discuss passive ion channel by discussing the resting membrane potential. A cell that exhibits a membrane potential is said to be polarized. And there are three important concepts for the resting potential. First, the extracellular fluid or the fluid outside the cell, and the intracellular fluid, which is the fluid inside the cell or the cytosol, differs in ionic composition. The extracellular fluid contains high concentration of sodium and chloride, while the cytosol contains high concentrations of potassium and negatively charged proteins. Second is that the cell have selectively permeable membranes, which means that ions cannot freely cross the lipid portions of the plasma membrane. They can enter or leave the cell only through membrane channels. In addition to this, the cell's passive and active transport mechanisms do not ensure an equal distribution of charges across its plasma membrane because membrane permeability varies by ion. For example, it is easier for potassium to diffuse out of the cell through a potassium leak channel than it is for sodium to enter the cell through a sodium leak channel. Additionally, negatively charged proteins inside the cell are too large to cross the membrane. As a result, the membrane's inner surface has an excess of negative charges with respect to the outer surface. The third concept is that the passive processes acting across the cell membrane involve both chemical gradients and electrical gradients. And when we say chemical gradients, they are concentration gradients of ions like sodium and potassium. An electrical gradient is when charges are separated by cell membrane. The inside of the cell is negative compared to the extracellular fluid. Active processes across cell membrane is when ions move back and forth, One example for active process is the sodium-potassium pump, which is powered by ATP or adenosine triphosphate. It balances the passive forces of diffusion. Remember that diffusion is the flow of solute from an area of higher concentration to an area of lower concentration. When ions begin to flow across the cell membrane, the sodium-potassium pump will correct the flow of balancing it by ejecting 3 ions of sodium for every 2 ions of potassium brought in. In this figure, we can see the cell membrane and we also got protein channels. And in here, you can see a potassium leak channel and a sodium leak channel. You can also see ions floating around, and you can observe that there are quite a bit of potassium in the inside of the cell, but not very much outside of the cell. So the potassium leak channel is also known as a passive channel. The potassium leak channel is going to allow potassium to leak out, and remember that diffusion works by allowing ions to flow from a high to a low concentration passively. So if potassium is higher on the inside of the cell, by diffusion, potassium will flow out naturally. The same is true over the sodium leak channel. There is a high concentration of sodium outside of the cell. So according to diffusion, sodium is going to passively leak into the inside of the cell. As potassium leaks out and sodium leaks in, the dynamics of the resting cell will be changed. This is because potassium is more permeable to the cell membrane than sodium. So, potassium can easily exit the cell than sodium going inside the cell. In addition, negatively charged ions or molecules cannot cross the cell membrane, and this will lead to a more negatively charged inside of the cell and more positively charged extracellularly. As this happens, the sodium-potassium pump is going to correct the passive leakage by pumping 3 sodium ion out and pump 2 potassium ion in. The sodium-potassium pump uses ATP or adenosine triphosphate because it is working against concentration gradient. Why do the sodium-potassium pump needs to correct the passive leakage? This is to stabilize the resting membrane potential or to keep it at rest or keep it at negative 70 millivolts because that is the voltage of a resting neuron. In summary, the resting membrane potential exists because the cytosol differs from extracellular fluid in a chemical and ionic composition. In addition, the membrane potential changes in response to temporary changes in membrane permeability, which is a result of the opening and closing of specific membrane channels, which happens in response to a stimulus. We now move forward with the discussion of active ion channels, and there are 3 types of active channels, namely the chemically gated ion channel, the mechanically gated ion channel, and the voltage-gated ion channel. When we say chemically gated ion channel, this is also known as your ligand-gated ion channel. They will open when they bind to a specific chemical such as your neurotransmitter. So there will be an attachment of a ligand or an ion or a neurotransmitter in the binding site of the ion channel. And they are usually found on cell body and dendrites of neurons. Mechanically gated ion channel will respond to membrane disruption. So they are found in sensory receptors that respond to touch, pressure, or vibration. A voltage-gated ion channel, on the other hand, respond to changes in membrane potential. So they will open if there is a change in the membrane potential or in the electrical charge of the cell. They are found in axons of neurons and sarcolemma of skeletal and cardiac muscle cells. The activation gate will open when stimulated, while inactivation gate closes to stop ion movement. So here's a picture of a chemically gated ion channel, and on the right side you can see that the ion channel is closed. If a chemical like a neurotransmitter, which is represented by acetylcholine in this picture, will bind to the ligand-gated channel, the channel will open and sodium will rush inside of the cell. When the neurotransmitter is no longer attached or present, the channel will close and sodium will not be able to come in any longer. The mechanically gated channel only responds if the cell membrane is disrupted. So here, you can see that the channel is closed. Once a mechanical stimulus such as touch, pressure, vibration, or tissue stretching is present, this will cause the channel to open and sodium begins to rush in. If the stimulus is taken away, the channel will close. The voltage-gated ion channel will be controlled by membrane potential changes. The picture in your right, as you can see, is at rest because the voltage is at negative 70 millivolts. If there is stimulation, the membrane potential changes. The gate, which in this picture is a voltage-gated potassium ion channel. It will open and potassium will rush outside of the cell. Please do take note that once we reach a certain membrane potential, for example at positive 30 millivolts, which is the highest voltage point, the voltage-gated ion channel will close or it will be inactivated. Neurons communicate with one another using nerve action potential, which is also known as your nerve impulses. They propagate changes in membrane potential and it can affect an entire excitable membrane. It will begin in the initial segment of the axon, which is at the axon hillock. Below is a picture of a neuron and you can see the axon hillock. Just below the axon hillock, it is the initial segment where the action potential or the nerve impulse begins and it will propagate towards the synaptic terminal. The generation of action potentials depends on the existence of a membrane potential and the presence of voltage-gated channels for sodium and potassium. In the previous slides, I've mentioned that a graded potential is the temporary localized change in resting membrane potential which is caused by a stimulus. The action potential is stimulated by a graded potential that depolarizes the axolemma to threshold. The threshold or the minimum requirement for an axon is negative 60 to negative 55 millivolts. It means that if we want to have an action potential, we need to at least have the membrane potential to change to negative 60 or negative 55 millivolts. When we say depolarization, it is when the membrane polarization is changed from negative 70 millivolts, which is the resting membrane potential, to negative 60 millivolts up to positive 30 millivolts, which is a result of the opening of the voltage-gated sodium channel. And then when we say repolarization, the voltage-gated potassium channel opens and the membrane potential goes back to the resting membrane potential, which is negative 70 millivolts. An action potential works by an all or none principle, meaning that any stimulus that changes the membrane potential to threshold will cause an action potential. A threshold is when an action potential occurs in the membrane of the axon of a neuron when depolarization reaches a certain level, which is mostly at negative 55 millivolts. All action potentials are the same, meaning that no matter how large the stimulus is, an action potential is either triggered or not triggered. That is why it is called all or none. And then when we say refractory period, this is from the beginning of an action potential up to the end, or when the membrane returns to the resting state. During this time, even if a stimulus is administered, another action potential cannot be generated. In order to generate an action potential, first, we need to depolarize the neuron. And depolarization, as we talked about earlier, it takes place when a resting membrane potential, negative 70 millivolts, changes towards a more positive number. A stimulus will cause depolarization to threshold. Remember that we need to get at least negative 55 millivolts, and once we hit that, we are going to activate depolarization to continue. Afterwards, sodium channels will open, then we are going to have full depolarization. That is when sodium rushes into the cytosol and the inner membrane will change from negative to positive. So the inflow of sodium changes the membrane potential from negative 55 millivolts to positive 30 millivolts. At the peak of the action potential, the inside of the membrane is positive 30 millivolts, more positive than the outside. At positive 30 millivolts, there will be an inactivation of sodium channels and activation of potassium channels, causing potassium ions to move out of the cell. This will shift the membrane potential back towards the resting level. This is what we call repolarization. We return to resting membrane potential as we get to negative 70 millivolts. The voltage-gated potassium channel will begin to close, and as the membrane reaches the normal resting potential, the potassium ion will continue to leave the cell, and the membrane potential may or will drop to negative 90 millivolts. And this is what we call hyperpolarization. After all the voltage-gated potassium channel finish closing, the resting membrane potential will be restored. Charge will go back from negative 90 millivolts to negative 70 millivolts, and the action potential is over. Remember, refractory period is from the beginning of an action potential up to the end when the membrane returned to the resting state. And during this time, even if a stimulus is administered, another action potential cannot be generated. Conduction, also known as propagation, is the movement of an action potential along an axon in a series of steps. There are two types of conduction, the continuous conduction and saltatory conduction. Continuous conduction occurs only in unmyelinated axons. It affects one segment of an axon at a time. It occurs as a step-by-step process along an unmyelinated axon. In continuous conduction, action potential develops at the initial segment. A stimulus will cause the sodium channels to open and sodium will rush inside the cell membrane, making the inside of the cell more positive than the outside. So the membranes will be depolarized to positive 30 millivolts, and then a local current will develop, and that local current will spread the stimulus to the next segment. This time, the next segment will be depolarized. So what will happen to the initial segment? There will be repolarization of the initial segment. Remember that at positive 30 millivolts, there will be an inactivation of sodium channels and activation of potassium channels, causing potassium ions to move out of the cell. This will shift the membrane potential back towards the resting level. Saltatory conduction, on the other hand, occurs in myelinated axons. This is faster than continuous conduction and requires less energy. The myelin prevents continuous conduction, and local current jumps from one node of Ranvier to another. In saltatory conduction, depolarization occurs only at the nodes of Ranvier. In saltatory conduction, again, a stimulus will cause the sodium channels to open, making the membranes to be depolarized to positive 30 millivolts. The local current will develop, but instead of spreading segment to segment, it will only depolarize at the nodes of Ranvier. So myelinated axons will be skipped over.